It's a pleasure to introduce our next speaker, who's Stephen LaConte. He's an associate professor here at the VTCRI and the Department of Biomedical Engineering and Mechanics at Virginia Tech. Stephen has published over 40 papers in his very young career and was selected as a Caplet Fellow of the American Academy of Sciences. Steve?
Thank you very much. Thwarted by cables. We need wireless. Okay, great. I'd like to actually just jump into the talk and start with a simple thought experiment. What would it mean if we could find localized regions on the surface of cortex that let us track deeper brain structures, including distributed networks and subcortical regions? Well, in one sense, this would be tremendously convenient because the surface of the brain is, relatively speaking, very accessible, navigatable, and amenable to recording. But it would also be disturbing in a sense because this small patch of cortex would then be serving as a redundant repeater signal of other processes that are happening in the brain. But if we relax this thought experiment a little bit and allow ourselves to develop models of cortex, of the surface of cortex, that could then do exactly this, then we can take advantage of the fact that the cortex is highly connected with itself as well as the rest of the brain. And so my group has been looking at this problem in terms of developing fMRI models restricted to the surface of cortex to see if we could track localized regions as well as distributed networks. Now, in this first simplified case, the work involved for modeling this would be fairly simple. We would have no to just a small number of parameters that we'd need to fit. But if we then open up the question that we're looking at on distributed surfaces of the brain, then we need a multivariate weighting function throughout the brain to help us reconstruct these signals. And so one principled method that my lab falls back to time and time again for developing these multivariate weights is machine learning techniques. And so this then becomes the umbrella organizing the talk, which is applying machine learning to fMRI data. And I should mention that many groups do this type of analysis for many different purposes. Our usual motivation is to build machine learning based real-time fMRI experiments from these tasks. So I'm going to talk about three types of ongoing work that we've been doing for quite a while, which is task-based prediction of fMRI, predicting resting state networks, and predicting localized regions. And that will set the stage then for this question of, can we actually restrict our models to the surface of cortex and come up with good ways to track arbitrary regions and networks? So jumping into task-based fMRI, if we just ignore this top arrow for a moment, the basic setup is that we have a human in the scanner, and we're usually asking them to do some sort of stimulus-guided task. And the machine learning goal of this is to come up with classification or regression models that first of all explain the data that we're seeing in terms of brain images and the stimulus or task parameters that are happening at the time these images are recorded. And then once we have this model, then we can add this blue arrow back in and look at things like neurofeedback, adaptive fMRI experiments where parameters of the stimulus are changing based on ongoing processes of the brain, or we can think about using this as a platform for studying brain-computer interfaces. So in a longer talk or a different talk, I'd be showing lots of different movies of task-based projects that we've done in the past, but suffice it to say, across a number of different interfaces and weightings. By the way, these maps are derived from these multivariate weightings that I was talking about, which help you actually distinguish which parts of the brain are most helpful in distinguishing tasks, such as very simple left versus right finger tapping, to perhaps more nuanced enhancing craving versus suppressing craving in cue reactivity studies. One commonality throughout all of these, though, that I want to mention is that this process is very robust. So in a matter of just a few minutes, we can have a working neurofeedback system. And we can use this to drive these computer interfaces, but we can also drive interfaces in the outside world as well. So this is just a, this is a robotic hand that was designed and printed on a 3D printer here at Virginia Tech by a team of mechanical engineering students. They came up and they were interested in neuroprosthetic types of applications. And so as just a first step, we explored the idea of using different tasks to create different hand signs in the American Sign Language alphabet. And so the master student that was actually part of the team that built this hand was actually in the scanner testing this out. In the interest of time, I'm not going to play very much from this movie, but this is me in the scanner that's in this building driving this robotic vehicle in a building that's on the main campus of Virginia Tech. So I'm getting on a live video stream, and I'm trying to follow the graduate student who actually built this robotic vehicle without crashing into expensive equipment that they have in their lab. So this is a wide range of tasks, but one common thing that we've found is that across all of these tasks, whether or not the person is controlling an interface or not matters for our modeling. So in particular, if we're classifying data that occurred when people were controlling a feedback interface, we tend to get a boost in classification accuracy. And this seems to be caused by an increase in whole brain signal to noise and facilitated by a frontal parietal insula network, which we think is implicated in this. Okay, so what happens when we take away that task that we have people do in the scanner and we just let them rest in the scanner and do what they do? Well, unfortunately, this has been dubbed resting state experiments by the field, even though everyone acknowledges that the brain is not at rest. But these networks are important because they've been replicated very broadly across labs worldwide. And so there are a number of groups that have replicated resting state patterns such as these. This particular set comes from a large meta-analysis of task data as well as independent component analysis of resting state data. And many of these networks have names like salience network or default mode network. But in our minds, at least, the precise functional roles of these networks are not clearly defined. So as an example, the default mode network is probably the hallmark of all of these, and it's well known that this network is suppressed when people are engaged in difficult tasks. I guess speaking for me is a difficult task today, so my default mode must be really low as I'm trying to do it. But we know that the DMN is also modulated both high and low by a variety of different tasks. So how can we reconcile the disparity or breadth of roles in which this network is implicated? And in our minds, at least, we think that real-time fMRI can be used to design experiments to tease apart what these functional roles of these networks are. So the trick here is that we no longer have task conditions to use as our labels for this machine learning method. So we have to actually extract the fluctuations, the highs and lows of the default mode network from the data themselves. Then we can train a machine learning model that acts as a regression-based predictor in future data. So not to overwhelm you with a lot of redundant data, but these are just individual traces from one run of resting state data where we used one run as a training run and then predicted the time course of the other run. So you can see doing this in two, we just swap the roles, and we get very high prediction accuracy. And I'm not cherry picking these data because these are across a number of subjects. We see very different time series, but fairly consistent goodness of fit, if you will, across these time series. And you don't need to train on resting state data and then test just on resting state data. You can actually go back to task data. So you build your model from resting state data, and then you can track things like the default mode network. So in this case, we had people do a fairly difficult executive function task, and we bookended it with a really simple task, which is just staring at a fixation symbol. So we go from very easy, doing nothing, to making some very simple responses. And so we see the DMN goes from high to low. And then when we go to the harder task, we keep that DMN low, and then we go easy, hard, easy, hard, easy, hard, and then super easy again. And if we look at the literature in terms of the default mode network as it applies to neurologic and psychiatric illness, it's implicated in virtually everything that it's been looked at. And so we've actually examined this in our own data of longitudinal traumatic brain injury, and we see that this anti-correlated nature of the DMN with executive function tasks is restored over time, starting from acute time points after concussion to several months later. So if we believe even half of the literature on the implications of the default mode network, one application that we're very keen on is, can we actually train individuals to rebalance their default mode network by using neurofeedback? And perhaps, if this is the case, individuals will enjoy broad cognitive benefits from this sort of therapy or treatment approach. The other application I've mentioned is that we're trying to tease apart the exact functional roles of these networks and actually put them to hard tests. And so this experiment was motivated by a growing literature that looks at the coordination of frontal-parietal networks with the default mode network in terms of vigilance, attention, commission of errors. And so essentially what we're doing is we're triggering some of these number sequences to occur without the subject actually realizing that their brain's frontal-parietal network is controlling some of the sequences that are appearing on the screen. And the idea is that if the literature is correct, we can actually push around performance on this task to make people either better or worse at a target detection task. Okay, so the idea for doing localized region prediction is very similar after kind of explaining both task-based and resting state networks. The idea of reconstructing a time signal recording from one part of the brain using other parts of the brain goes back to electrophysiology data, but the first example that I found in the neuroimaging literature has come from Carl Friston, actually using PET data, and he was interested in doing this to look at LTP and LDT. Coincidentally. But the idea is that if you're interested in some region of interest in the brain, can you exclude it from your model but use the rest of the brain to try to reconstruct the time series in these data? So we actually set out to characterize how well this could be done, and a postdoc in my laboratory, former postdoc Cameron Craddock, came into my lab already having developed a parcellated atlas of the brain, and so we actually applied that. So in this case, we wanted to track posterior cingulate, or what I'm showing is that we're tracking posterior cingulate, and these then are the multivariate weights that help us reconstruct these images. So this is the best subject, and this is the worst subject in this cohort. And we went further and looked at every parcel and essentially ranked each region of the brain in terms of how reproducible, here in panel B, those multivariate weights were, and how well we could predict those time series up here in prediction accuracy. And going further, I don't know if my timer is running. Okay. Okay. I'm blazing through because I know that lunch is after my words. Okay. Great. So we've actually we actually think that there are some organizing principles here that we need to go after further. But when we look at both the prediction accuracy across the brain as well as the reproducibility of these multivariate weights, we see that there is some, perhaps some leads here to go after these organizing principles. Okay, so now going into kind of where we started. Can we actually predict networks and regions with surface-based models? And we get this surface using just very simple morphometric image processing techniques, starting from a whole brain mask of brain versus non-brain. And not surprisingly, we have a monotonically increasing ability to predict regions and networks the thicker we make our mask. But for everything I'm going to show in subsequent slides, we picked a thickness of 10 millimeters because that's a fairly good guess at where we could be creating recordings using fNIRS optodes at typical optode spacings and optical flux power. So just looking at this, again, this is the last time series I'm going to show, but here is a network from that Smith level template. Turns out that this happens to be a very easy time series to predict because part of the network itself is defined by the surface of the brain. But here we see the network predicted value, the actual network fluctuations, and our predictions of this value using the Hokie maroon and the Hokie orange as well. And so to kind of summarize all the time series that we were looking at, I'm using these bar graphs. So we looked at amygdala, anterior cingulate, caudate, insula, posterior cingulate, and putamen, as well as those 10 resting state networks that I showed earlier. And there is quite a bit of variability in terms of how well we can do this, but I want to point out that everything that we've looked at is statistically significant and withstands multiple comparisons correction as well. So given this in a fairly small cohort, I wanted to look further at what regions of the brain, what these multivariate weights were telling us about what was going on to support these predictions. So we actually trained models on 99 subjects and did a group analysis which could show us what was consistent across individuals in terms of tracking these different regions and networks. Let's see if this is slow. So I can't fit all of them easily on each slide, so we're just going to scroll down. And then just to kind of, it's a lot to actually digest, and so this next format may not help that much more, but I thought I'd just take a pause to look at amygdala, for example. Basically what we're showing here is the multivariate weighting on average across these 99 individuals that help us to reconstruct this amygdala signal. And if you pour through, as we've been trying to do, decades of literature that involves cat and monkeys and as well as human functional connectivity, these maps are actually quite plausible. And so here is just another example of posterior cingulate. Again, these are group results from 99 subjects. So the individual subject models may have quite a bit of variability, but you can see that these summarize fairly nicely. So then related to posterior cingulate then would be the network models of the default mode network. And so I've decided to show that here. And then a very interesting one, at least in my mind, are these left and right frontal parietal networks. And to me, they're interesting because of both their symmetry and asymmetry that you can see here. Okay. And then without going into a lot of the detail, this was kind of the easy case scenario where we used the entire cortex and the full resolution of fMRI, but we wanted to look at a closer simulation of what we would get when we start to move this into other technologies such as fNIRS. And so what we've done is we've taken regions that have shown up in these group maps of the 99 individuals and basically used an algorithm to try to move them into more optode digestible patches that we could get through a dense fNIRS array. And so going back to the original burnt orange predictions using the entire resolution of the surface from fMRI to which included thousands of voxels to to just the patches, which would include dozens of voxels on average for these regions and networks, we see that we do take a hit in our prediction accuracy, but they still are statistically significantly above chance, and again, this survives multiple comparisons correction. Okay, so I've covered a lot in a pretty short amount of time, but to just kind of recap, predicting task-based fMRI provides a highly flexible experimental tool, and we're finding that the neurofeedback actually enhances classifier performance, and we think the human performance of the task is what's actually supporting this. Predicting resting state networks is something that we're really going after in terms of applications, because we think that this could be a very unique, holistic approach to rehabilitation after stroke or TBI, for example. But really, if you look at the literature, this could be very broadly applicable in a lot of domains. And we want to take these resting state networks that people are basically using as templates, and we actually want to put them to an experimental test that's harder than just a correlative report. We want to design experiments that take advantage of our theories about what the functional roles of these networks are, and see if we can design tasks that people can actually use to modulate networks, or we can use them to, say, enhance or suppress accuracy on tasks where we think that these networks are implicated. For predicting localized regions, we've begun to characterize the prediction accuracy and reproducibility of these regions, and I think we have more work to do, and I think that there's some promising underlying principles there that we should be looking at further. And then in terms of this latest idea of looking at surface-based regions, obviously the next step is that we're moving towards validating these findings with fNIRS. But what we think that the kind of motivating idea is that we think that we're kind of approaching a multimodal and algorithmic strategy to address the issues of spatial resolution and depth of penetration for other recording techniques such as fNIRS. So this is important because this allows us to take this very powerful but in many ways limited imaging technique of fMRI and move it to other domains that are cheaper, more portable, more shrapnel safe, quieter, all of those sorts of things. So can we actually leverage all the strengths of fMRI and move that into other techniques like EEG, fNIRS, MEG, for example? So I'd just like to thank my group, but in particular, Jonathan Lasinski has been involved in all aspects of basically everything I've shown today. Cameron Craddock, a former postdoc in the lab, was pivotal in getting the resting state network prediction working, as well as the localized prediction. And then Mark Tinzer has taken this idea and run with it for all of the surface-based results that I've shown. So I'd also like to thank my past and current funding, and thank all of you. And just before we go to questions, Tor almost broke out in kind of a beer drinking song, or a recap of a song he was singing this last night with Harry Sontheimer. And then in the panel discussion, we were talking about, whether or not people would want this invasion of privacy of recording of our brain activity. And so it reminded me of something I'd forgotten for a number of years. I was watching Mr. Rogers, and if you don't know Mr. Rogers, he's a very popular, especially when I was a kid, a kids show, and the extremely nicest person and interesting person in probably the world at the time I was a child. But there was an episode I was watching with my son many years ago, and he sternly was looking into the camera and said, not even the fanciest x-ray machines or MRI in the world could ever read your thoughts. I almost fell off my chair when he said that. What you're thinking is for you to know, Fred Rogers. So I thought I'd just kind of end with that.